All right, well, yikes, sorry. Whoa. Sorry, yikes. Sorry. Welcome back, everybody. Oh, my God. That's really bad. I'm sorry, everybody. That's surprisingly horrible. All right, let's see. Okay, well, now that I have your attention, today is Molecular Biology 3. There's a minute or so on the clicker clock. I want to do the next step, which is translation, how you go from RNA to protein. And at that point, you'll actually have almost every piece that you need to understand what went wrong and what went right when you were messing around with the Gene Explorer. Right? With the exception of introns, which we'll talk about next week, you'll have all the pieces. So I want to say a little bit about how it works. Translation is more complicated than transcription, but in the end, it works by base pairing. We'll talk about genetic code and do an example. There is no lab next week because Thanksgiving. Yay. So there is a warm-up due Wednesday morning, and there's some specifications on the final exam. Logistical or administrative questions before I get down to business. Oh, yeah, is there a lecture on Wednesday? Oh, yeah. Oh, yeah. Oh, there's lecture on Wednesday, yeah. Right, I mean, unless there's an earthquake or a flood or a volcano or something. I am so sorry. I'm scared of my microphone now. Why is it doing that? All right. Well, we'll see how we do. All right. For Christ's sake. All right. Fourth, we'll count on the clicker question. When is exam 3? Exam 3 is it 2 weeks from Monday? It's December 5th. So I think it's 2 weeks from Monday. All right. So the right answer was B, which is correct. Good job, everybody. Yes. All right. That's an important analogy. Oh, man. All right. So let's get down to business. Translation. And like I said before, transcription, translation, they sound a lot alike, and it's easy to goof them up. But they're really quite different things. And so we want to keep them straight. The way like I said, I remember transcription is easy. You're just copying over. Translation is actually a more challenging thing, like translating from English to French or something. So today I want to talk about how that works. As a reminder, that's making protein based on the mRNA sequence. So to be clear, oftentimes we'll say we're making protein from RNA or turning RNA into protein, and that's not what we're doing. We are reading the RNA and using that information to generate a protein. It's not a chemical conversion, it's a reading operation. So in the same way we talked about it before, the mRNA and the protein, you made a little table. mRNA has 4 letters. How many letters in the protein alphabet? How many amino acids do we worry about? A lot, right. It turns out there's 20. I'm really sorry about the noise. There's nothing I can do about this because if I take this off, you can't hear me. So I'm sorry about the huge explosion sounds. Hopefully they will go away. There are 20 letters in the protein alphabet. They are, in mRNA, it's A, G, C, and U. And here it's ALA, VAL, LYS, etc. 20 different ones, it turns out. They're one strand in RNA and one strand in protein. That makes things a little simpler. This goes from 5' to 3', and this goes from amino to carboxyl. Right? A couple things. This is done by a thing called a ribosome. Okay? And that is a complex of protein and what's called rRNA, ribosomal RNA. So if you were going to be consistent in how you named it, you'd call it protein polymerase. Right? The thing is, it turns out, historically speaking, they discovered ribosomes long before they knew what they did, because they're actually quite large as cellular organelles go. So there were these things called ribosomes. Ribo because it contained a lot of RNA, ribonucleic acid. We now call it the rRNA, the ribosomal RNA. Later on, they figured out that those things are actually where proteins are made. So they have a sort of archaic name. The other thing is, because there's a different number of letters, the translation can't be as simple as transcription. Transcription, it was just every time you see an A, you're going to pair a T, etc. Right? That's easy. One for one letter. In this case, you've got a 4-letter alphabet you're trying to turn into a 20-letter alphabet. So you can't read one for one. So therefore, you need to read the mRNA in groups of nucleotides. These are called codons. Okay? And you can actually do a little bit of math. And this is one of the kind of cool things. The number of nucleotides per group, that is per codon, okay, and the number of amino acids that can be specified. Right. And this must be at least 20 for all 20 amino acids. Right? So if you have one nucleotide per codon, you can specify 4 different amino acids. You could have an amino acid for A, one for G, one for C, and one for T. That's only 4 amino acids. That's nowhere near the 20 that you need. So what about if you had 2 nucleotides per group? Well, then you could have a G, a C, and a U, GG, GC, GG, and GU, etc. If you work out the math on that, it's 4 times 4 is 16 possible amino acids. Both of these are too few. They're not enough for the 20 amino acids you need. If you, on the other hand, you do 3, it's 4 times 4 times 4 is 64, which is more than enough. And this is one of those cool things. Lots of times biology is just so. It's the way it is. You have to live with it. It doesn't always make sense, but that's the way it is. This is one of those few things where it actually makes sense. That is the smallest number, the smallest possible size for a codon that can encode all the different 20 amino acids is 3 nucleotides. And it could have been that they're 27 long, but there's lots of extra, right? But it turns out, in this one particular case, 3 is exactly right. That is, 3 is just what you need, or it's a little more. It gives you more than enough capacity, but that's the way it is actually in real life. Therefore, there are 3 mRNA nucleotides per codon. And again, it didn't have to be sort of as neat as that. And when the Watson and Crick and all their buddies were working on this, they thought, wouldn't it be cool if it was 3? The math would work out kind of nicely. And it turns out that it does. It didn't have to, but it turns out that it does. So the other thing you need, so as we'll see, CCC encodes proline. You need some way to go from the RNA world, CCC, to the amino acid world. And people had lots of weird ideas back in the day that they thought perhaps that the RNA had a particular shape, so only the right amino acid would plug in. Except you can't make that work. It just won't, the shapes aren't right, and they're not versatile enough. So you had to have something a little bit different. And this was actually proposed by Francis Crick of Watson and Crick. He proposed that there were going to have to be an adapter. So you also need a nucleotide to amino acid adapter. All right, because all these things work by base pairing. So you've got to have some kind of molecule that has one end that can base pair and another end that has an amino acid hanging off of it. And so Crick said you're going to have something like that, and they went and they looked, and it turned out they found it. This is the thing that's called tRNA, or transfer RNA. And there's about 64 different kinds. right, because of the 64 different codons. It's slightly more complicated than that, but that's not a bad place to start. And if you look at what a tRNA looks like, let me give myself a little bit more space. If you got your mRNA, 5' end out here, da 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 da, and you've got a little bit of sequence, da 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 da, to the 3' end. Say you have A, C, C. That's the codon you want to read. Well, your tRNA has to base pair that. It's going to have a G, G, U. And in fact, for honest-to-God base pairing, this end has to go off to the 5' end. These things have to be anti-parallel. And this tRNA molecule is base paired to itself in very complicated ways. And then at the 3' end, there's an oxygen atom, and there'll be the amino acid attached. I'm going to do this in red. Okay. And so this whole thing is the tRNA with the specific amino acid attached. Okay. And the idea is that, so for every codon, the cell has to make a separate tRNA, in fact, many copies of the same molecule, with the ability to bind to it the 3 nucleotides that encode that amino acid, and hanging off the other end is the actual amino acid itself. This is an adapter between the RNA world and the protein world. So how does translation work? Base pairing, right? Because you got base pairs down here. I'll draw them in some color. Let's do it in green. There are base pairs. So one end of this molecule base pairs with the RNA, and the other end with the codon, and the other end has the amino acid in question attached. Questions about that guy before we talk about details of how the whole process really works? Okay. So the other thing you're going to need, like all these processes, also you need a start and stop signals, like transcription. That is, all of these processes have a start signal and an end signal, so the thing knows where to do its job. Okay? And that was the analogy thing that we talked about for the clicker question. So what I want to do now is take you step by step through the process, and then we'll talk about details. So the process looks something like this. Number one, the ribosome starts at the start codon. We'll talk about what that is in a second. Start closest to the 5' end of the mRNA. So the mRNA is read 5' to 3'. Ribosome grabs on the end and rolls along, grabs on the 5' end and rolls along until it sees the first start codon. We'll talk about that is in just a sec. And then the tRNA with the proper amino acid base pairs with the mRNA, the messenger RNA. And then number 3, the amino acid is added to the protein as the chain grows, and the mRNA is read 5' to 3'. The protein is made amino tarcarboxyl. So that's why I made a fuss about the amino to carboxyl direction, because we usually write protein sequences amino terminus to carboxyl terminus because that's the direction they're actually made, the way they're synthesized, and how they'll line up with the RNA. So we keep our sequences aligned. And then the ribosome advances 3 nucleotides and this process repeats. And you basically repeat that until you run into, at the stop codon, mRNA and the protein are released. And as a note, the protein folds as it is made. Okay, so I'll show you an animation in a second, but what I picture is, I don't know if you guys ever played with Play-Doh when you were a little kid, those sort of fuzzy pumper things, you know, you push the lever and the Play-Doh comes squeeging out. Well, that's kind of what happens. So the ribosome squeezes the protein out. And as it comes out, it folds up in all those ways we talked about, hydrogen bonds, ionic bonds, hydrophobic interactions, etc. Right? What I want to direct your attention to is there in today's information-packed handout, there's a bunch of still frames from some of those animations. And I'm going to show you an animation just for a second to remind you where we are in the process. And then I want to show you an animation of translation to sort of make this process a little bit clearer. So give people a chance to write that stuff down and find the handouts, and then I'll play the movies. Okay. So this one. So this is our general central dogma thing. We start with DNA because that's what genes are made of. Right? The first step was what we talked about on Wednesday. You peel the strands apart, and you're going to copy one of them into mRNA. You're going to transcribe one. And this shows a strand going away. It doesn't really go away. It's sort of ignored for the moment. Right? Because remember, transcription is not trying to copy everything, just this one gene of interest. So it copies this one gene into mRNA, transcription. We talked, like I said, about Wednesday. How does it work? Base pairing, right? Base comes in, base pairs and joins, base pairs and joins. Why does it not make the wrong sequence? Because the wrong base won't base pair. And then the RNA gets peeled off. And what this doesn't show is the DNA is then going to reform. The DNA helix will zip back up. The RNA comes out of the RNA polymerase. And then the ribosome chunks along, reading them in groups of 3, that's what we're talking about now, and makes the protein. And those are the amino acids, one amino acid for every 3 nucleotides in the RNA. So that's the sort of big picture of where we're at. Now, if we want to zoom in on translation, this animation is a bit more complicated than we needed, but it contains various important pieces. So this big thing is going to be the ribosome. It turns out the ribosome assembles out of many big pieces on the RNA. So first of all, some pieces come in which you will not worry about, initiation factors, they call them, to start the transcription process. All right, so here you got the RNA. All right, so the backbone and the various bases sticking out, single-stranded. This doesn't show the start codon very well, but it shows how the process basically works. So what happens is, this bad boy up here is the tRNA. Why is it all coiled up like that? It's like a protein, right? It folds up. It's a single strand of RNA, but what makes it fold up is not hydrophobic interactions like we're used to. It's actually base pairing. It doesn't show it here, but these loops are held together because this is an RNA and has nucleotides sticking out, and they actually base pair with themselves, and it gives it a simple structure based entirely on base pairing. And here, this is what's called the anticodon. It's going to pair with the RNA by regular base pairing, and that's the amino acid. And there are enzymes in the cell that ensure that the right amino acid gets stuck on the right tRNA. So this is going to come in, and how does the right one come in? Basically, the one that base pairs is the only one that can stay, so it comes in and sticks by base pairing. The ribosome assembles, and then one by one, new tRNAs come in. And this makes it sort of look choreographed, like only the right one comes in, right? But in fact, lots of the wrong ones come in and they get bounced out because they don't base pair. Only the one that can base pair properly will stay. And then there's sort of a very subtle detail. I wish this animation was a little bit lighter. But the thing to watch now, here's the amino acid from the first tRNA. Here's the amino acid on the second tRNA. There's going to be a switcheroo as we assemble the protein. They transfer over, shoop, and now you've got a protein with 2 amino acids, and a new tRNA has come in. And you go along, it shoops along, the 3rd amino acid gets added. So that was the first one in the chain, second one, third one, fourth one. So amino terminus, et cetera, et cetera. You add more and more RNAs. And at each step along the way, let me back it up 1 second. At each step along the way, the empty tRNAs come out. So ones that are called charged, ones with amino acids come in. Once they've dropped their amino acid off, they're spit out by the ribosome, and they get recycled. So the enzyme, the proper enzyme comes along and sticks a new amino acid on that so it can go around again. And so then this thing goes sort of chugging along, 3 nucleotides at a step, adding amino acids to the protein one by one. Base pair and join, except it's 3 at a time now instead of 1. And then it rolls along doing its thing. And then what they do is they zoom out, and what's interesting is you can actually have several ribosomes reading the same RNA, kind of one after the other. The one at the front will have the longer protein because it's been on longer, and then the ones behind it will be less far advanced. Let me back that up and let it play one more time. Questions about what's going on here? Yeah. Why does the protein? Why does one amino acid get joined onto the other? So there's 2 ways to look at why that happens. First of all, if you didn't do that, you wouldn't make a protein, right? But then what the ribosome does is do some little bit of chemistry to join those amino acids together so that they become a protein chain. So because remember, the protein chain is a bunch of covalently bonded amino acids. You actually got to nail them together with a covalent bond. And so that's where the peptide bond gets formed is like right at that instance, that little sort of, right, if you look in, right up in there is where that peptide bond is going to form. And so, oink, there wasn't there, and now, ping, now they're bonded together, right? And so now you've got a two amino acid chain, then you'll have a 3 and a 4. Other questions? Yeah. Yeah. Yep. Where do the tRNAs get the amino acid from? That's a cool question. There are enzymes which recognize the tRNA, and they can read the anticodon and know, oh, this is the right one, and then they stick the appropriate amino acid on. So for each tRNA, there's actually an enzyme whose job it is to stick the right amino acid on. Yeah. Does the ribosome pause on each codon? Yes. That's right. As far as I know, it kind of sits there, does its thing, and then goes kerchunk 3 at a time. Kerchunk. That's right. It will not move unless it gets the proper amino acid. That's absolutely right. So, for example, if you don't have enough of suppose you have to put a tryptophan in there, and you don't have enough tryptophan, the ribosome will stop and wait for a tryptophan tRNA to come in. Right. Okay. Yeah. Oh, yeah, the beginning, they show there's 2 colored balls going at the beginning. There are protein factors that get the whole thing started. We won't sweat about them, but the ribosome is actually a very complicated thing. Other questions? Yeah, Raquel. So what happens to the tRNA after it loses the amino acid? It gets recycled. It gets recharged. The enzymes grab it and stick the proper amino acid back on again, so it can get reused. Right. All right. So that takes care of that, takes care of that. Okay, cool. So what I want to direct your attention to now is the actual genetic code. So if you go to the next page of today's information packed handout, you'll see something like that. And so there's a couple things to notice. What does this tell you? Every amino acid has this is the codon and the amino acid. So if there's an AUA in the mRNA, that encodes isoleucine, ACA, threonine, etc. So this is a table. And if you count them up, there's 64 different codons. It shows all the possible combinations of 3 A, C, U's, and G's. And it lists out the 20 amino acids. All right. So there's a couple things to know about this. Number 1 is this guy. Actually, let me do this in color. This is the start codon. All right. It's AUG, and it starts translation, and also encodes an amino acid, methionine. All right, that's number 1. All right. Now, there are also 3 stop codons. And what this says is stop translation, and there's no amino acid added. All right? So, before we go further, let's take a second and look at this. There's some other things about this too. You have 20 amino acids, 64 codons. There's going to be some duplicates. So take a second. There's two things. I want to look at with your neighbor, and I'll call on people's names. Which amino acids have the most codons for them? And there's one amino acid, or there's a couple of amino acids that have a very few number of codons. So take a minute and look at the table. Find me an amino acid that has a very large number of codons for it and one that has a very small number of codons. So talk to your neighbor, and we'll call names and get started and do all that. All right. So whoever I call, you get to pick. The first person gets to pick whether you're doing a large number or a small number. But all right, so get a consensus on one or the other, and we'll call a name. So Kyle, Sweezy, Kyle, tell me, sir, you want a large number or a small number? What do you want to do? For large, tell me, what's got a lot of codons? Alanine has a lot. How many? 4. All right, so if there are many, like alanine, thank you, sir, which have 4 codons. So it's redundant. This is 4 codons. We can do better than just 4. Yeah. So bigger one, go for it. Arginine. Yeah, Arg has got how many? 6, right? So Arg has got 6. These 4 and these 2, all right? So Arg has got 6, all right? There's another one out there that's got a lot. Yeah. Leucine has got 6 also. There's a lot that have 4, a decent number that have 2, all right? How about the small numbers? Let me give a chance to give somebody some credit for this. How about a small number? Adrienne, are you here? Adrienne, tell me, something's got a small number. Methionine has got only 1, okay. How about another one that's got only 1? Yeah. Tryptophan, right? So that there's some that are only 1. So thank you, Adrienne. Let me do that in a different color. So the green one, this one has only 1. And Trip has only 1. All right. So, all right, that happens to be the way it is. Questions about that before we go one more step? Yeah, in the blue. Why that some have more and some have less? It isn't clear. For a time, it was thought to be sort of an accident. It actually turns out that some folks, I'm doing a little bit of this research myself, other people have done lots of research on this. There's reason to believe that this is actually not an accident, that it's actually evolved in a way to make the effects of mutations smaller, that the code is laid out in a non-random way. At this point, it's not obvious why. The reasons are fairly subtle, but there's a chance that it's not random. At the first approximation, there's no particular reason. It turns out leucine is one of the most common amino acids in proteins and it has a lot of codons, and tryptophan is rare, but arginine is also rare, so it's not really clear. Yeah? Why is there 1 start and 3 stops? Again, I don't know. I don't think it is known. It's also another really good question. Yeah? Wait, let me talk about that in 1 sec. All right. So what he asked, because I didn't want to give this away, is my next question is, given the rules about start and stop codons, what can you, and this is a talk to you and everything, what can you tell me about the beginning, the amino terminal amino acid, of every protein on Earth, if anything, and what can you tell me about the carboxyl terminal, the last amino acid, the first and last amino acid in every protein in the world, is there anything, given what I've just set up here, you can say about either or both of them? Can they be any amino acid, or is there some restriction? So can it be any amino acid or some restriction for the N-terminus, the first one, and can there any restriction on what would be at the C-terminus? So think about it, talk to your neighbor. Given what I said about the way start and stop codons work, is there anything you can say? All right, so get a consensus. We'll get, and the first person I call gets to pick, start or end. All right, so let's see. Drew Rishardi. Drew, are you here? Drew, so tell me, what do you want to pick, the start or the end? So tell me about the start then. Is there any restriction on it? It will always be methionine. Absolutely. Drew's got it right on the money. Because the start codon encodes methionine, every single protein is born with methionine at its N-terminus, as you asked. That's exactly right. Because a start codon happens to encode an amino acid, every protein starts with met. That's the truth. All right, that's the story. All right, so therefore, all proteins start with methionine at their N-terminus. Okay? All right, so what about the C-terminus? Let me pick somebody. Sophia. Sophia, are you here? Sophia? Oh, there you are. Hi, Sophia, sorry. So can you say anything about the C-terminus, the end of the protein? No. I see you said no very quietly. That's absolutely right. It can be any amino acid. Because basically you roll along, you start with a start codon, methionine, alanine, leucine, blah, 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 blah. You put the last amino acid in. It can be anything. And then you put a stop codon. And then it stops. So she's absolutely right. You can have any amino acid you like at the C-terminus. So thank you. So, and can end with any amino acid at the C-terminus. Okay. That's kind of big, so I want to make sure there are no questions about that. But it's a direct consequence of the way the code works. Okay. I want to draw a little cartoon of this process, and then I want to do an example, because that's the best way to really make sure that you understand what's going on. So here we go. So an example. Right? A simple gene. Okay? So the gene itself is, as always, DNA. And there's a 5' end, and a 3' end, and a 5' end, and a 3' end. All right? And you're going to have, at one end, promoter. Talked about it last time, that says start making RNA. And at the far end, you're going to have a terminator. Okay. And then what's going to happen is transcription is going to make your mRNA, which extends from just after the promoter to just before the terminator. 5', 3'. This is the mRNA, as it would have been shown in the GeneX. And that also has, somewhere along here, a start codon, which is not the same sequence as a promoter, and a stop codon down here, which is not the same sequence as a terminator. And then the next step, I'm out of cool colors, I guess I'll just use blue. Bless you, translation makes a protein. And it's shorter because it was 1 amino acid for every 3 nucleotides in the RNA. So that's your basic sort of cartoon of how the gene does its thing. A couple things to bear in mind, which are sort of counterintuitive at first. Remember, we're not transcribing all the DNA, just the gene we care about. So there's DNA on either end that we are not transcribing. We are not even translating all the RNA. There are typically leaders and trailer regions with a little bit of RNA that's not transcribed. I mean, why not have your start codon right at the beginning? And why not have your stop codon right at the end? The answer is, from the point of view of Bio 111, there's no explanation for that. In real life, the sequences before the start and after the stop are involved in regulation, controlling when this gene is expressed and in what cells and in what parts of the cell. So it's actually, in real life, it's important, even though it's not obvious at where we're talking about. Questions about that before we do an example? Okay, so if you turn to the next page of today's information packed handout, it will look something like this. So putting it together. Right? So you got a DNA, you got a promoter, you got a start of the RNA, blah, blah, blah. First question, talk to your neighbor. Given that information, are you going to make RNA A or RNA B? We'll talk protein in a second. This is sort of a review from last time. Which RNA are you going to make and why? Why can you only make one? The same drill. So think about it, talk to your neighbor, or call names. If you make an RNA A or RNA B, and then I'll call on somebody. We'll hand out a fair number of points today. And for what it's worth, this is like exactly a classic exam problem. There's lots of, exam 3 will almost certainly have a problem that looks a lot like this. Bless you. Bless you again. All right. So get a consensus, and let me call a name to report for the group. So, Taimi, I think I'm saying that right, Taimi? Are you here? Yes, no, yes. Did I say it right? Taimi, sorry. So tell me, mRNA A or B? Yes, you certainly can pass. Passing is totally fine. So pass. All right, let me do one more name. Tom. Thomas Stetson. Thomas, tell me, A or B? I got to vote for B. All right, let's see. All right, let's see if we can make that work. Thank you. So if it were B, which strand would it be paired with? If you're making B, it's got to be paired with the top strand or the bottom strand of the DNA? With the top strand. If it's paired with this top strand, can that legally be paired with the top strand? No, because it would be going 5' to 3' in the same direction. So it cannot pair with the top strand. So let me write that out to be really clear. It cannot be B. So this would pair with top strand. But the DNA is going 5' to 3', and you'd try to base pair with an RNA going 5' to 3', and this is a no-no. This is not, they are not anti-parallel. And Tom, I'm actually super glad you said that, because I'm sure you're not the only one, and this is one of those cases where showing why the wrong answer is wrong is way more important than knowing the right answer. But let me be clear. Is everybody with me why this cannot be right? Questions about why that cannot be right? Yes? So the answer is, in order to be the sequence CCCG, that means it has to pair with GGGC, right? So that means it's pairing with the top strand. Except you can't pair with the top strand because to make a bona fide base pair, they have to have strands have to go in opposite directions. So this is an illegal structure. All right? Yeah.
So if the promoter region is on the 5' at the top,
Yeah.
does that mean that it cannot bind the answer A either? Because in order for it to bind answer A, the promoter region would have to go to the 5' at the top.
No, so let me be clear. The promoter region, because your question is about what strand, hey everybody, what strand the promoter's not on one strand or another. It's actually that whole sequence on both strands. So let me put it this way. Probably a safer way to think about it is A is the correct answer. Let me do that in a different color. Because this is going to be paired with bottom strand. Bottom strand. And that's a legal base pair. So you've got mRNA going 5' to 3'. And the DNA on the bottom strand is going 5' to 3' that way. La, la, la. And this is okay. It follows all the rules. Right? Questions about that before we do the next stage? Yeah? Yep. Let's leave. If you want to talk about protein, I want to postpone that. Because I heard you. Let's postpone that for 1 second. I want to make sure everybody's with me on the RNA part because this is really important. Yeah. Oh, no, that's not a question. Yes. Paired with the paired. No, that will pair with the bottom strand because G pairs with C. All right. So if you draw it out, you'll see. Yeah. Oh my goodness. I did write it wrong. Holy mackerel. Yes, all right, sorry. Oh yeah, I totally meant to do that so that you guys would check it and make sure that you had it right. Yeah, right. All right. Sorry, I did have it wrong. No wonder with all this like muttering in the room. All right, fine. Okay, yes, I'm sorry. Yikes. Okay, so that is correct. All right? Yes. So you can see how easy it is to mess these things up. All right, very well. Let us move on to translation. The next question is what protein is being made. And I want to zoom in on that RNA. So I've got this thing zoomed in here. So what I'd like to do the same thing. Talk to your neighbor. What is the amino acid sequence of the protein that's getting made here? So follow the rules. Think like a ribosome, if you like. What's the protein sequence going to be? So think about it. Talk to your neighbor. I'll call names. All right, so get a consensus, and let's walk through this guy. All right, let's see. Gabrielle, are you here, Gabrielle? Gabrielle. Oh, all right, Gabrielle, tell me, what's the first amino acid? And so you say methionine. Are there any takers for GGG glycine as the first amino acid? A few, all right. So remember, think like a ribosome. You don't start at you start at the beginning, but you roll along until you see the first start codon, which is AUG. So GGG is not a start codon. GGC is not. GCA is not. CAU is not. I'm getting kind of bored here. AUG, oh my goodness, I found a start codon, right? So Gabrielle's got it, right? So the first amino acid is always methionine. So here's the start, right? And so the amino terminus, H3N plus met, right? And then, what's the next amino acid? Proline, that's CCU. We can put in commas here. This becomes pro. Then, what's the next one? Gly, comma, right? And then UUG, leucine. I'll trust you. I don't remember the code all that well. AUG, methionine. Wait, do we have a problem here? Yeah. We have a problem. Yeah. Well, we just put a start code in the middle of things. Turns out that's just fine. You can have methionines in the middle of a protein, right? How does the ribosome know what to do? It starts at the 5' end and finds the first start codon. So there's no problem. It doesn't try starting here because ribosomes, they grab at this end and roll until they see the first start codon. Right? So you can have methionine in the middle. It's OK. All right? CGG, arginine, UGA. Stop. All right, now here's the thing. The right answer to write here is COO minus. Let me write that in carefully. This is the stop codon. All right? So I have a thing. If on an exam you write stop as the amino acid, I'll take off tons of points. It makes me crazy, right? Because anyone can look at that table and go, oh, stop, the STOP amino acid. Right? That you don't have to know any biology to do that. Biologists know that there is no STOP amino acid, right? That's the end of the chain. So that's where the carboxyl terminus goes. So if you write STOP as an amino acid, bad things will happen to your grade. I'm sorry that I have this fetish about that, but I just do. I know we're almost out of time, but there's a couple points I need to make here. I have a question that I want to ask you. If you look at this here, what does UGA encode? It's a stop codon. How come the protein doesn't stop there? It's out of frame. All right? So when is a stop codon not a stop codon? When it's not in frame. This is not a stop codon because it's not in frame. What do I mean by in frame? The commas. Hey, everybody, this is important stuff. How do you know where the commas go? Every 3. Do they start from the very beginning every 3? No. They start from the start codon in every 3. So does there have to be a multiple of 3 nucleotides at the very beginning? No. It can be any number. The commas are set by the first start codon. Sorry, so let me just say it. So that the reading frame, the commas, or the reading frame is set by the first AUG. All right. Let's do the clicker question because we're almost out of time, and then we will call it a day. Answered. All right.